Hello everyone, in this video we're going to talk about meiosis, but first a quick advertisement for my trip to the Smoky Mountains. We have 10 people already signed up and we have 5 more spots open for students who want to potentially sign up. So if you think you might be interested or if you know someone at school who might be interested in this trip, please let them know and they can talk to me in the next few days. So let's get started with mitosis, because part of meiosis is very, very similar to mitosis. So if you can remember mitosis, it's about regular cell division. The function of it is for asexual reproduction or for growth and repair of the organism that do sexual reproduction, right? So if you think about people, we also do mitosis in our body a whole lot, way more than meiosis actually, because we need mitosis to make more cells to allow us to grow and repair every single day. And for both mitosis and meiosis, we can talk about the parent cell. So this is not just the parent, but a parent cell, right? So if you think about sexual reproduction, you would have 2 gametes coming together, but each gamete comes from one parent. But right now we're talking about one cell, not the reproduction, but the process of mitosis, that's starting one cell. And then what you end up with are 2 daughter cells, but both parent cell and daughter cells are generally diploid. But if you have parent cells that are haploid, you can also have daughter cells that are haploid. But either way, mitosis gives you the same exact cell as the parent, but now you have 2 copies of it. So if you look at the stages of mitosis, we have metaphase, well, prophase first, metaphase, anaphase, and then telophase. During prophase, just real quick review, and then cytokinesis. All right. So during prophase, you can see the chromosomes, and then metaphase, the chromosomes line up at the metaphase plate. Anaphase, the sister chromatids, wait, what did I say? The sister chromatids line up at the metaphase plate, and then they split during anaphase. And then telophase, the nuclear membranes reform. So one more time, you can see the chromosomes now. Metaphase, sister chromatids line up. Sister chromatids split. And then nucleus reform during telophase. And then cytokinesis, there are 2 types. There's the cleavage furrow for animal cells, and then there is the cell plate for plant cells. So make sure you understand all of these aspects. One quick note for prophase again. So during prophase, that's the first time that you can see the chromosomes, the condensed form of the DNA. You cannot see the DNA that are condensed during interphase. So remember, our cell cycle goes like this. We have G1, S, G2, M. So during G1, S, and G2, which are interphase, our cell prepares for division, but during this time, you can't see the chromosomes condensed yet because we're not ready to split up the chromosomes yet. All right, now we can look at meiosis. So make sure that because mitosis and meiosis, they don't really come hand in hand per se, but they're very similar processes, and those processes happening in your body. That's why we need to, these are some of the reasons why we need to think about both processes at the same time. So here's mitosis, where there's only 1 division, but meiosis has 2 cell divisions. That's why we go from 1 cell, 2 cell, and then 4 cells, okay? So here's mitosis. Meiosis, cell splits once, and then each cell split again. Now we have 4 cells, and we can count the chromosomes as well. 46, 46, 46, 23, and 23. So the first step of meiosis, we would already go from diploid to haploid. And then the second step of meiosis, meiosis 2, we go from haploid 23 to haploid again. So let's look at, well, this video, you can watch it, but let's look at this picture right here. It's a little bit more clear, and it's not moving as fast. So here's meiosis. This is where we want to focus on. During prophase, we have diploid cells. Diploid means for each type of chromosome, you would have a pair of it, okay? So it's kind of like you would have, let's say we have pens, right? We can have 3 different kinds of pens. We can have pencils, we can have regular pens, and we can have markers. But for pencils, we can have 2 pencils of different types, right? Whether they're both pencils, but one could be number 2 pencil, the other one could be number 4, if that's a real thing, right? But they're both pencils. They're kind of very similar, but they're 2 versions of the same pencil. And then the second one, you can have pens. Let's say we have big pen. You can have the red big pen and the blue big pen. But they're both big pens. They're kind of like twins. In the third pair, you could have 2 markers. You can have 1 black marker, one red marker, but they're both markers. So what's the point of this? These two will be called homologous chromosomes because they're coding for the same traits. Okay? So how this works is, let's say this part of this homologous chromosome codes for your eye color red for some reason, and then over here at the same locus, which is the location on the chromosome, you would have the same exact gene. So whenever you have a same gene, that means this gene is coding for the same product. It's either the same protein or the same trait, okay? You want to repeat after me. The definition of a gene is really important, at least for my class. The definition of a gene is a sequence of DNA, all right? We have DNA right here, DNA right here. A sequence of a DNA, part of the DNA, a sequence of DNA that codes for a protein or a trait. So the gene that's coded right here, or the gene that is at this part of this chromosome, it's the same gene right here, which means it codes for the same trait. So if this is eye color right here, this is eye color right here. This is hair color, this is hair color. If this is your, what else has a color? Your skin color. This should be skin color right here as well. Okay, so that's homologous chromosome. But then if we were to have another pair of homologous chromosomes right here, they will be coding for the same, for different things, right? So earlier we had eye colors already. So right here you would have, let's say, length of eyelashes as well. Widow's peak as well. Okay. So these are called homologous chromosomes. They're a pair of chromosomes that are coding for the same genes and the same traits, but they come in pairs. It doesn't mean that, well, this could be black eye and red eye, but then you can also have black eye and black eye together as homologous chromosomes, but they're homologous chromosomes, okay? So during prophase 1, again, we said that's the time. So you need to know the events that happen within each one of these phases, okay? So first off, with prophase 1, DNA condensed into chromosomes, so the X shapes. You don't have to say the X shapes. That's just to help you remember how they look like. So DNA condensed into chromosomes. Homologous chromosomes will pair up. So this chromosome right here and this chromosome will pair up. And then these two chromosomes will pair up. Crossing over happens. And I'll talk about what crossing over is in a little bit, and then nucleus disappears. All right, so let's count the events. We have DNA condensed into chromosomes, homologous chromosomes pair up, crossing over happens, and then nuclear membrane disappears, or nuclear membrane fragments, okay? 4 important ones. During metaphase, homologous chromosomes line up at the metaphase plate. So first off, you should know what a metaphase plate is. It's this imaginary region in the middle of the cell. There's no real plate there. That's just where the homologous chromosomes line up. And you should know what homologous chromosomes are. But this, if you look right here and right here, this is called homologous chromosomes line up. This is called sister chromatids line up. As you can see, they look very different from each other, right? Homologous chromosomes are lined up because you want to split the homologous chromosomes. As anaphase, homologous chromosomes split. Telophase, the nuclear membrane reform around where each cell is supposed to be, the DNAs. And then eventually cytokinesis split the one cell into 2 cells. So the important part right here is you can see we started off with 2n equals to 4, meaning diploid number is diploid because there are homologous chromosomes in there. And then 4 means how many total chromosomes are there. There are 4 chromosomes in total. So 2n equals to 4. And then at the end of meiosis 1, you would have n equals to 2. N means now each one of our cells is haploid because there's no more pairs of chromosomes. They're all individual chromosomes. And 2 means within each cell you would have 2 chromosomes. Okay? So this is meiosis 1. If you're not exactly clear on what each step is and you don't remember what's going on, please watch a video. Okay? Please, please, please. This is on the test for sure, and you have to understand it very clearly. Now we can move on to meiosis 2. At the beginning of meiosis 2, this is right after meiosis 1, meiosis 2 would happen, okay? So at the beginning of meiosis 2, each cell already have haploid number, in this case, n equals to 2 for human, n equals to 23, Remember how you can count the chromosomes. One X shape is one chromosome, but one line is also one chromosome. So right here we have 4 chromosomes, 2n equals to 4, 2 chromosomes in each cell, n equals to 2. So during metaphase 2, the sister chromatids line up at the metaphase plate. During anaphase 2, the sister chromatids separate from each other, being pulled by the spindle fibers. And then telophase, the nuclear membrane reform around the chromosomes. Cytokinesis, the daughter cells are formed. So now, as a result of meiosis, we would have 4 different cells. Each one is haploid. So 4 haploid daughter cells that are different from each other, and they're different from the parent cell. Okay? So this right here. N still equals to 2. It just right here, n equals to 2, and these are duplicated chromosomes. Right here we have n equals to 2, and they're unduplicated chromosomes. So meiosis is also called a reduction event because we're actually reducing the number of chromosomes from diploid to haploid. Okay. Now, just a few more things. One thing is called crossing over. That's an event that happens during meiosis. The result is that it increases genetic diversity for the entire population. So here's how this works. During prophase 1, we have our homologous chromosomes pair up, right? But because they're so close together, some part of the homologous chromosomes can switch parts with each other, as you can see over here. So the genes that's located right here and right here, they are the same gene, but they might be different versions of the gene, right? And same as here, and same as here, and same as here. On every DNA, there's a ton of genes. So when crossing over happens, the versions of the genes switch so that when meiosis allows homologous chromosomes to split and then allows each sister chromatid to split, that will give you daughter cells that have very distinct genes or versions of the genes or alleles left in the cell. So if you look at this picture right here, right, this is all blue, all pink, but now you can have part blue, part pink right here, right here, and right here, and each of the daughter cells is not the same. But if crossing over doesn't happen, then you'll have 2 daughter cells that are the same as each other, and the other 2 are also the same as each other. Now, this next concept is called independent assortment. Independent assortment happens because homologous chromosomes can line up in any possible way during the metaphase, okay? So you can have homologous chromosomes line up like this, and then the next pair could line up like this or this. The next pair can line up like this or this. So for every single pair of homologous chromosome, you would have 2 possible lineups, this or this. Why does it matter? Because if you were to have blue eye coded on this homologous chromosome and green eye coded on this chromosome, right? This will allow this daughter cell that are formed to have blue eyes, and then these daughter cells to have green eyes. But if you were to switch it, and then the next one, for the next gene, let's say hair color, also switches, or this way, that will give you different combinations of every single trait as a result of independent assortment. So let's see. If we were to only have 2 pairs of homologous chromosomes, how many chromosomes are there? 1, 2, 3, 4. So 4 individual chromosomes. You would have, during metaphase, you can have this line up, right, where these 2 blue chromosomes are separated together and these red chromosomes are separated together, and you'll have these 2 combinations for the daughter cells. But then for you could also line up like this, where you would have these 2 different combinations for the daughter cells. So this increases genetic diversity, which means every single gamete that's produced by a male or a female is different from each other as a result of independent assortment and crossing over. And this is great because that's what makes everybody different from each other, and so that's what allows us to keep on evolving and then to survive in the environment. The last one is called self-differentiation, to something that we mentioned before. Stem cells are undifferentiated, unspecialized cell, which means they don't have a specific job and they don't have a specific, function and structure quite yet. But then stem cells can differentiate into specific types of cells that have very distinct shapes and structures and functions and location in the body. So that's it about meiosis and self-differentiation. Please ask me if you have any questions. I will be at school in the morning at the library.